Now we're going to talk about multicellularity and first, the adaptations of animal cells that make it work. Before we do that, let's say, why bother with multicellularity? And these points that I've listed here are four equally valid ways of looking at it. One is to consider the division of labor, the subject from last time, the extreme specialization of motor nerves and muscles and the way they mate together to enable control of bodily movements. That's a really extreme example of the division of labor. Perhaps a simpler one might be, well, why not have some cells specialized for, say, motility and some for digestion? So perhaps the poster child for this viewpoint is this humble animal, trichoplax, which is a flat little plate of cells. Some people imagine that it's something like what the original animals might have looked like. It's basically got four different cell types, and it's more or less differentiated into a ciliated outer layer and or upper layer, and then a lower layer specialized for digestion. Presumably, as it crawls along, it picks things up and eats them, and then interfaces with the fluid world through a ciliated layer. Again, some people imagine this is perhaps what the simplest original animals looked like. It's equally likely that it's basically just someone's liver that ran away and decided to make a go of it on its own. Okay, whatever. Second point of view, water control. I mentioned in the very first lecture in this series that many of our epithelial cells bear some sort of relationship to the choanoflagellates, a structural and functional relationship to the choanoflagellates that are our closest living unicellular relatives. So this kind of kidney-centric view of multicellularity focuses on the propensity to define an outside versus an inside and control the microenvironment. We might nominate as the poster child for that sort of thing something like, I'll just draw a sea urchin blastula, which has an epithelium, a ciliated epithelium that's out there swimming around. And one of the first things that happens during sea urchin development is that some cells leave that epithelium to invade this internal fluid cavity, which is different from the outside world. Whereupon, as in many animals, they go on to differentiate to make organs that are protected to some extent from the outside world, although they, of course, must exchange with it. Third point of view, of course, multicellular organisms, one of the main motives in the history of life for adopting a multicellular lifestyle is for the sake of motility or size or general body form. And instead of us, let's just say, let's nominate something like this familiar organism for the poster child of point 3 here. That's supposed to be something like a fir tree, okay? If it weren't for multicellularity, life on planet Earth would be a bunch of scums and plaques, okay? Finally, fourth viewpoint has to do with this division between the germline and the soma. And I'm going to return to that with an illustration from another multicellular group in the next lecture. So let's take as a starting point to consider some of the cellular adaptations for multicellularity. Let's take a family of related differentiated cell types, muscle. We've talked about striated muscle, or so-called skeletal muscle. Within each muscle, like let's say one of your big ones, a bicep or something like that, there are a bunch of bundles, fascicles. Those bundles group together these giant multinucleate myotubes, within which are these highly ordered contractile bundles, the myofibrils. Then we have invertebrates, cardiac muscle. This is a very distinct muscle type, but it shares some features with striated muscle. Cardiac muscle is not a multinucleate tissue. Cardiac muscle cells are uninucleate. They're stapled to each other tightly by cell-to-cell junctions. Like skeletal muscles, they contain very ordered arrays of contractile units, all oriented the same way. You'll forgive me if I resort to a really iconographic style at this point. Cardiac muscle shares a lot of features with striated muscle. The contractile units in particular are well ordered. They have a very well-defined rest length and maximal extent of contraction. The difference is simply that these are uninucleate cells that are bound by tight junctions, and they are electrically coupled to each other such that a calcium rise in one triggers a calcium rise in the neighboring cell. These excitably coupled tissues work together. These cells in an excitably coupled tissue work together to produce a regular and rapid contraction, much like striated muscle does. Smooth muscle, like cardiac muscle, is individual cells. Smooth muscles, for example, line our vasculature, make up many of the muscles of other organs and tissues. Let me put that off to the side for convenience, actually. And also the bodily motility of many invertebrates. In smooth muscle, the contractile units are not nearly as ordered. Fibers might span the cell in many orientations, and furthermore, they do not have as regular a periodicity of contractile units, although there is still some pseudosarcomeric organization to actin and myosin in smooth muscle. Another major difference is that smooth muscle is not contracting rapidly in response to something like the action potential, although calcium has a role it has a role in stimulating the contractility of myosin more directly by phosphorylation. The Rho GTPase also stimulates myosin activity in smooth muscle cells. Smooth muscle cells can achieve, over a much longer time scale, they can achieve a greater extent of contraction than either of these muscle types can, and they can often sustain it for quite a while rather than relaxing soon after. And finally, there are, at the other end of the scale, myoepithelial cells. Embedded in many epithelia, a subset of cells might exhibit the development of a long contractile process Where might you find such a thing? Well, as far as I'm aware, essentially all cnidarian muscles are myoepithelial. Students taking the lab have seen myoepithelial cells in, say, starfish embryos and larvae. These contractile extensions in myoepithelial cells are even less well organized than the smooth muscle cells. In fact, if you watch myoepithelial cells work in, say, a starfish larva or under the epidermis of a cnidarian of some sort, these strands don't even necessarily contract all along their lengths at once. Different parts of these strands can contract to different extents for different durations, as if the control of myoepithelial contraction is quite local indeed. That's very good, perhaps, for making small-scale tissue deformations, whereas, obviously, at the other end of the spectrum, striated muscle is very good for making powerful swinging motions of whole appendages. From the point of view of multicellularity, there is another very important distinction along this spectrum. There doesn't seem to be any major reason why this cell, this myoepithelial cell, could not return, could not de-differentiate and divide. It's a uninucleate cell. Perhaps it can retract its this specialized contractile apparatus, go through division, redifferentiate, etc. But striated muscle is an entirely different matter. Constructing it involves cell-cell fusion on a grand scale. There's no way that these cells are going to butt off from that great big thing and ever divide again. However, muscles and other tissues, of course, but we're going to talk about muscles as a representative example, they need repair. They might get damaged. And here we meet, in the context of the skeletal muscles of vertebrates, we meet the original stem cells. Hanging out alongside muscle fascicles are little nests, not by any means drawn to scale, because of course, if this is your bicep, that's something, a foot or so long, whereas these are supposed to be little tiny things. Those are what are called satellite cells. Hanging out within the fascia that bounds a muscle. In response to damage, satellite cells proliferate and make new replacement muscle cells. Stem cells, wherever they occur, are slow dividing cells. These satellite cells can't just be proliferating all the time. or they wouldn't serve the function that we're about to describe. They've got to hang out there, divide slowly until something calls for them to produce the precursors of, in this case, new muscle cells. These satellite cells have a single possible fate. When they divide, one daughter has to remain a satellite cell. It has to remain a stem cell. The other daughter goes on to proliferate, etc., until there are enough of them to produce a new muscle fiber. So this phase here, self-renewal, is one obligate part of a stem cell's job. Again, it wouldn't be a stem cell if it couldn't self-renew like that and maintain the reservoir of precursors. This phase, amplification, is the other part of its job. Finally, differentiation. Ultimately, these are supposed to fuse into the multinucleate myotube and differentiate myofibrils and so forth. Okay, we'll leave it at this rough level of description. This is a kind of stem cell. These satellite cells that regenerate skeletal muscles. This is a kind of stem cell that has a single possible fate. Okay, you might say it is unipotent. There is a general distinction when we talk about stem cells between pluripotency versus totipotency. And we could invent the word unipotency. Doesn't sound very good. This is a spectrum from, on one end, the ability to make a single differentiated cell type, or at the other end, the ability to make any differentiated cell type. If you've heard of embryonic stem cells, which people use to clone mammals, those are an example of a totipotent cell type. You can take mouse embryonic stem cells and inject them into the blastocyst of a mouse embryo, and the descendants of those cells will make anything possible to make in a mouse. In between, most stem cells are pluripotent. A good example is the hematopoietic stem cells, which give rise to all of the various cells of the blood. So it's hard to draw the hematopoietic case. Compared to striated muscle, a less restricted sort of stem cell population maintains your gut epithelium. A very simplified depiction of the anatomy of the small intestinal epithelium includes projections, villi, not microvilli. These are villi as in, I guess, macrovilli, fingers, versus crypts, that is, intrusions that go the other way. The villi include absorptive cells and secretory cells, all of the things that make the intestine work on a day-to-day basis. Those are all non-dividing cells up in the villi. They have a relatively short lifetime. They're sloughed off every few days to a week. The intestinal epithelium is turning over faster than most of the other tissues in our bodies. The crypts include the stem cells anchored at the base down here by what are called Paneth cells. So zooming in a little bit, the stem cells of interest that maintain the continuous growth of the intestinal epithelium are located down here in the crypt. This zone here is the so-called Paneth cells. They have a number of somewhat poorly understood roles, but among other things, they may make a niche that sustains the stem cell population, that maintains whoever happens to stay in that zone as a stem cell, whereas the ones that get out of that zone into this proliferation zone these are incidentally often known as transit amplifying cells. Fancy way of saying the ones that are actually doing the dividing to proliferate to make bigger numbers. And then, of course, this zone here is differentiated. So think of the intestinal epithelium as a kind of a continuous conveyor belt in which proliferation of some fraction of the stem cell daughters continually produces new differentiated intestinal epithelial cells of the various types that cohabit there, which perform their function for some time before being sloughed off. Signaling within the crypt promotes stem cell division in response to demand for new cells. So that's an example of a pluripotent, not totipotent, stem cell type. Let's not forget about other multicellular eukaryotes. Here is a rough diagram of the fundamental organizational plan for vascular plants. This here represents a section of stem. That's a leaf, the basic above ground organ of a vascular plant. And this part here is the axillary meristem. Axil means pit. Whereas up at the tip of the shoot, directing and sustaining shoot growth, is the shoot apical meristem. The vast majority of the cell division in a plant body as it grows takes place within the meristem. All the rest of it is basically cell expansion due to the vacuole. Inflation of the vacuole pressurizes the cells and makes, say, the stem grow and the leaf expand and things like that. Why so much terminal differentiation in multicellular organisms? Why restrict proliferation to small populations of cells? This is a very common theme across the eukaryotic world. Why is it so? We'll return to that question later. For now, we'll continue with some of the mechanics of multicellularity. It's not enough to just divide up the chores and then say, okay, kids, stick together, pull together, not apart, right? Okay, first of all animals developed from a blastula. Let's recall what that is. It's a starfish. It might look like this, cilia sticking out, columnar epithelium. Now, in some other animals, it might have fewer cells. Say, if we were looking at what passes for the blastula in a gastropod, it might have some big blocky cells instead, and relatively few of them. Nevertheless, or if we were looking at an insect embryo, say, it might be stretched out around an internal yolk mass or something like that instead of an internal fluid cavity. Nevertheless, they all have in common that the cells making up the blastula have acquired polarity. They've acquired junctions with each other. We blow those up, as we did before at the beginning of the class. Animal cell junctions include several types, some of which we've mentioned before. For example, animal cells often make attachments to some kind of extracellular matrix through what we call focal adhesions. Focal adhesions couple the cytoskeleton inside the cell in various ways to proteins of the extracellular matrix crossing the membrane. Other kinds of junctions couple cells to their neighbors in animal tissues. These include, so we've already mentioned focal adhesions. In vertebrate tissues, there are so-called tight junctions. Many invertebrates have the functionally equivalent septate junctions. As far as we're concerned, this is a distinction without a difference. The junctions between epithelial cells in the intestinal epithelium are so tight that not even water passes readily. That requires special proteins, but the one I really want to pay attention to is the so-called adherens junctions. And the reason is that adherens junctions are both in development and also perhaps in the evolution of animals, probably primitive. That is, they're the thing that come first. They're the thing that holds the cells of the blastula together. And the adherens are dependent on a set of calcium-dependent adhesion proteins called, because they are calcium-dependent adhesion proteins, cadherins. These are homophilic cell adhesion molecules. Homophilic means they bind to each other. A cadherin expressed on one cell binds to a cadherin of the same type expressed on a neighboring cell, and cadherins are often the basis for tissue type identities, that is, for the recognition within a tissue by the individual cells that compose it of cells of that tissue. There are several different types, E cadherins, N cadherins, etc. Not as many types as there are tissues, but at any rate, they differ in their adhesiveness. Again, they're calcium dependent, which is one of the reasons why most animal embryos, although not mature tissues like, say, your liver, most animal embryos can be dissociated into individual cells by putting them in calcium-free media. If we take one of these epithelial cells and look at it in sort of pseudo three-dimensional view, we might see that it has a polygonal apical end where the basal body is, that there are vertices I'm not going to draw the backside for hopeful clarity. Vertices around this polygonal entity, and that these junctions, which may or may not be concentrated at vertices, are associated with a cytoskeletal belt, predominantly based on actin filaments, but also other cytoskeletal participants in some cells. This so-called zonula adherens extends as a kind of a collar around an epithelial cell. It doesn't matter if we're talking about a columnar epithelial cell or a squamous one like this. Okay, this belt of actin-enriched cytoskeleton associated with the so-called zonula adherens, which just means, I guess, the zone of sticking together, adhering, is a dynamic entity that can undergo contraction or remodeling in various ways. Of course, none of the cell shape changes, none of the coordinated cell shape changes that characterize tissue development, the development of organs and things like that, would be possible if the cytoskeleton and all of these junctions between cells held together too well. They must, by their very nature, be relatively dynamic, turning over continually so that they can be remodeled and enable what we call morphogenesis, that is, patterned cell shape change. So what's in the repertoire here? Given a bunch of these that stick together, all connected, what can they do to make tissue shapes? Well, here's a really simple one. Got a few blocks like this. Well, somebody could decide to leave, right? Some cell, having acquired some sort of special state or a cue of some kind, could leave the epithelium. An example of this is the mesenchymal cell populations that students in the lab would have seen in echinoderm embryos and larvae. They undergo an epithelial mesenchymal transition, which is reversible. That is, mesenchymal cells can also join epithelia under certain circumstances. Here's another seemingly trivial cell shape change that we could find many uses for. Columnar epithelial cells could change their aspect ratio to become squamous, and vice versa, right? Now, it's fairly straightforward to imagine how this row could do that. In order for these cells to become squamous, this imagine that this is some sort of an elastic band around the zonula adherens, it would have to relax, widen, or, contrary-wise, in order for these cells to become columnar, perhaps a band around the margin of these cells, imagine them connected by neighbors, or connected to neighbors, rather, could constrict, changing the aspect ratio of these cells. This is not as trivial a behavior as it seems. For example, in the same echinoderm embryos, it is cell-shaped transformations like this that sculpt out of the initially uniform blastula wall a ciliary band, where the cells are tall and skinny to concentrate cilia together, versus the rest of the epidermis, which becomes quite thin. Choreographing that in a patterned way actually creates the ballooned out shape of the larval body. Okay, next, given a bunch of connected epithelial cells, they could buckle locally Oh, I hope I got the same number in there. 9 in one, 8 in the other. Okay, I didn't mean to make any cells divide there. Okay, when this blastula undergoes gastrulation by invagination in echinoderm embryos, when part of this epithelium dents inward, some number of cells must be coordinating a change in shape in a two-dimensional sheet, not just in a one-dimensional row, to mediate that buckling. One might imagine several ways that one could do this. We'll get back to that in a second. The fourth characteristic cell shape change that I want to diagram, now I'm going to look down upon a two-dimensional surface of epithelial cells. Let's imagine some cells that are roughly packed together hexagonally. That's the closest possible packing arrangement. And in real tissues, they aren't quite hexagons. There's some pentagons and heptagons in there. Now let us imagine that these cells start to exhibit polarized protrusive activity. Let's say that at each vertex that faces in a particular direction, cells start to wedge their way between their neighbors, that is, trying to crawl across one another. Let's look at this one on the top. Because I haven't drawn in any neighbors, it's obvious to see what's going to happen if it tries to crawl across its neighbor, right? It's going to extend a flap that way and a flap that way. Now, keep in mind, I said, let's imagine polarized protrusive activity. That is, so that these cells somehow know how to do it left and right as opposed to just any old where. If this cell continues to crawl across its neighbors, it will eventually, say, stretch it out in that direction like that. Having crawled across their neighbors in turn, these cells might have, let's say, met in the middle. This has the important and non-trivial consequence that the neighbor relationships within here are changing. These cells could continue to do this, of course, and end up crawling their way into a long stack instead of whatever it was they started as. You'll have to forgive me for the unintentional size change in the profile as I do this. That's not meant to be happening here. It's a byproduct of my myopic perspective up at the front here. Okay, This particular choreography is so widespread and useful that it has gotten its own heading used in, textbook indices and things like that. Convergence and extension, or convergent extension. What's the use of this? Well, let's draw that blastula that's just starting to gastrulate by invagination. Having buckled a sheet, maybe buckling it a little further during gastrulation. The challenge is now to elongate this tube, which is initially a stubby little cylinder, into an entire pipe. Well, one of the ways that this is achieved is by polarizing this kind of protrusive activity around this cylinder so that neighbor rearrangement within the cylinder forces the cylinder to elongate. This happens to be the way that in the embryos of ascidians and their vertebrate relatives, the notochord goes from being a plate of precursor cells to a single row of cells that then vacuolate to make the supporting skeleton for the muscular tail of a tadpole larva. Also, convergence and extension is the way many buds protrude into appendages. Combinations of these transformations, thus, as we've talked about in reference to the blastula undergoing gastrulation, let's not forget that this might be forming in particular zones the ciliated band as other parts of the epithelium thin out. Combinations of these transformations craft the epithelial linings of tissues and organs in all kinds of animals. For example, if you take number 3 and continue it for a while until you actually pinch off a tubule and then conduct convergence and extension within that tubule, then you'll make a long, thin thing appropriate perhaps for the neural tube, or perhaps a tracheal tube or something like that. Okay, so let's take a look at number 3 here finally, and consider there are 2 ways that you might buckle a sheet starting from this stereotype of a polygonal topped cell like this. Given this elastic waistband, imagine it going all the way around the back like that. It doesn't take much imagination to hypothesize that perhaps that waistband could constrict, and that in so doing, it might force some of the volume of the cell down. Indeed, this kind of transformation is often seen at the level of either whole groups of cells working together to create a buckling, or even individual cells that are undergoing this shape change within an epithelium. This is referred to logically enough as apical constriction. What else could we do? Well, an alternative hypothesis for cell shape change envisions that the cells within this epithelium all together might start to extend out that way. That as they do so, volume being a constant and all, there must be some sort of circulation. Imagine that each of these is kind of like an amoeba trying to crawl out that way, right? And that the cortex is continually protruding out toward the basal end and then circulating around toward the apical end. This so-called cortical tractor model, which is based on observations of the way cells really crawl, again, example being something like an amoeba with pseudopods, it's as if we imagine all cells doing this at the same time and staying stuck to each other as they do so. So that's another way to buckle a sheet like that. And believe it or not, there isn't that much more. There are a few other things I could put up here that are harder to draw, but actually this is a toolkit of morphogenetic movements that goes a pretty long way toward making an animal. There's one other key element that I want to highlight. And I'm going to start by describing a classic experiment on the nature of the plasma membrane, which involved mixing artificial liposomes. Liposomes are vesicles made of lipids, like the membrane lipids. And let's say we put one color of fluorescent dye on one population of vesicles and another color of fluorescent dye on the other population of vesicles. Recall, it was not always known to what extent the plasma membrane is a fluid. Well, one of the demonstrations that it is fluid-like is that when vesicles fuse, in this case, of course, initially, right at the instant of fusion, whether it's soap bubbles or membrane lipids or what, you're going to get half that vesicle labeled with one color and half labeled with the other color. But over time, the expectation is that diffusion will blend them. And the more liquid-like, the more rapidly that this will take place. Okay, seems like a trivial thing. The membrane is, however, therefore a fluid sheet, and diffusion blends lipids within it, and also proteins dissolved within it. Here's the thing. This isn't necessarily true for epithelial cells. Okay? You might find that cells maintain compartmentalized distributions of labeled lipids. In part, this is maintained by the existence of diffusion barriers in many epithelia associated with, say, septate junctions around the, the apical end. And also, I should mention, by the fact that there is continual circulation of membrane lipids through the endosomal pathway. If that's choreographed with a certain polarity to it, then, of course, that could contribute to the maintenance of two distinct compartments. The importance of this observation, however, is deeper than that because it implies that epithelial cells always have a distinct apical from basolateral end. Cells need some sort of polarizing cue in order to establish that. And the last piece I want to include in this particular lecture has to do with the one of the molecular kits, as it were, that enables that simple maintenance of distinct unmixed compartments within the cell membrane. The start of this story is with the zygote of the nematode C. elegans, which is elongated along its future anterior-posterior axis just after fertilization, and it looks terrible. So here's some polar bodies out here. There's a female pronucleus in one end, near where meiosis took place. Because of the way these eggs are fertilized, the male pronucleus is clear at the opposite end. Don't worry, they don't have too far to go. It's not that kind of exercise. These are tiny eggs. They're only about 50 microns long. This hideous look here turns out to be due to ruffling motility. And what's going on here is that everywhere on the cortex of these cells are little focal contractions, transient contractions that buckle the membrane in a little bit. This ruffling motility is soon polarized after the entry of the sperm. Something about the sperm suppresses that. Don't want to draw that quite so big. Something about sperm entry suppresses that ruffling motility, which continues in the anterior end, and in fact is driving a flow of material, a visible flow of material that way. This goes by the very logical name of cortical flow. At about this time, the pronuclei will be getting together back here. It happens that in C. elegans embryos, I should label that, this margin between the zone in which focal contraction continues and the zone in which it's suppressed, this margin actually ingresses quite a bit, almost like a cleavage furrow. It's called the pseudo-cleavage furrow. But that is a transient thing, and once the nuclei are have sidled up to each other in the center and they're starting to think about forming the mitotic apparatus, the embryo has become smooth, this ruffling has ceased, but in its wake, it leaves two distinct membrane domains. So this is the analogous partitioning of these marks. But in this case, the marks in question were identified by genetic screens as a series of cortical proteins. That characterize the two halves of this now highly polarized cell. On the anterior end, the proteins par3, par6, and a partner, a protein kinase, so-called atypical protein kinase C, are cortically localized. Whereas on the posterior, a different set of proteins, again, one of them is a kinase, are cortically localized. They have formed these two mutually exclusive zones representing approximately half the embryonic surface. So if we zoom in to some intermediate state in here where there's still partitioning, what's going on the plasma membrane is something like this. Par3 is a protein that tends to oligomerize. So I'm going to draw it as little hexagons here. And as it oligomerizes, it happens to associate with elements of the cortical cytoskeleton, actin-myosin networks. These oligomers of par3, since they've associated with the cytoskeleton, then as it flows, these things are moving with it. In fact, these aggregates are developing partly as a consequence of these focal contractions, it appears. Par6 is a little adapter protein, and it makes a complex with APKC, this protein kinase. And I'm just going to draw it so that it fits onto the edge of these par3 aggregates. That kinase targets a number of things, but one of the proteins it targets is the posterior pars. So if I draw the complex between par1 and par4, just iconically, I'll make it a triangle or something like that. This phosphorylates that complex and makes it fall off. There's a role here for CDC42, the Rho family GTPase. I'm going to have to diagram that. I used a Q before. I'm going to have to diagram that as an upside down Q. That also, CDC42 also recruits par6, which again phosphorylates these posterior par proteins and makes them fall off. The general idea is that CDC42 recruits these proteins to the surface where they meet these forming aggregates of par3, and then they ride along with them. CDC42 in this context also promotes contractility. So CDC42 is promoting the contraction that is dragging all these forward. This protein complex, the par1, par4 complex, remember I said it's also a kinase, and it has a reciprocal effect. That is, it inhibits the association of the anterior pars with the cortex as well. And because of their mutual antagonism, because one of them couples to the cortical flow machinery that is induced on one half of the egg, they divide the egg surface between them. Why does this matter? This was discovered in C. elegans by genetic screens looking for defects in embryonic polarization. But it turns out that the PAR proteins, in particular, the anterior PAR complex, these guys, are widely used throughout the animal world to polarize epithelial cells. It is the anterior PAR complex, now often referred to as the apical PAR complex, that defines the apical domain of epithelial cells.